to introduce you to the work we're doing at Anima Biotech. Our main area of focus is mRNA translation regulation, and I would like to use one or two slides to introduce you to this fascinating area in biology. Essentially, in our genes, we have all the features that dictate the regulation and fate of a protein that needs to be translated from that specific gene. Once a gene is transcribed, the regulatory elements that are included in the final RNA are essentially dictated during transcription, pre-mRNA processing, and finally splicing. 5' UTR length can be different in different mRNA isoforms that are produced from the gene by the fact that different transcription sites are being used, and different 3' UTRs can be utilized depending on where the poly A tail is introduced. This is called polyadenylation site usage, and there are genes that have more than one such polyadenylation site. At the end of the day, this will determine which cis regulatory element in both UTRs are being included, and this in turn is important for inclusion of regulation on the level of the fully processed mRNA. So at the end of the day, one gene can give multiple isoforms, not only because of splicing, but also because of different UTRs, that results in different docking places for RNA binding proteins or splicing factors, which determine the fate of the mRNA, how long the half-life would be, where it would be transported in the cells, and how well ribosomes will recognize it, and when will translation be initiated. This regulatory protein as a family is a fairly large family. It includes RNA binding proteins, ribosome accessory proteins, and enzymes that modify RNA, and all of these are regulated in a highly tissue-specific manner, which turned out that when we have these cis regulatory elements in the mRNAs, this mRNA's fate can be differentially regulated in different cells depending on the proteins that are expressed in a specific cellular environment, and determine the regulation of the pre-RNA into the mRNA. So just as an example, RNA binding proteins, there's about 1,200 of them that are encoded in our genome, and as a family, they're actually one of the most tissue selective expressed proteins that they are. So the same mRNA can be regulated differently depending on the RNA binding protein that is expressed in a specific tissue as cell type. Inside the cells, not all ribosomes are the same. There's accessory proteins and ribosome protein paralogs that essentially determine different ribosomes, and each one of these is responsible for the translation of a specific set of mRNAs that are coordinately regulated, and thereby we can interfere specifically in the translation of one or a selected biological pathway in response to a specific outside stimuli. In cancer, for instance, it is known that different enzymes that modify RNA are regulated or aberrantly regulated, and again, this changes the recognition sequences on mRNAs to these proteins that bind the RNA, thereby regulating the ability of these mRNAs to be regulated and translated by ribosomes. All of this, at the end of the day, is important for both selectivity and specificity in mRNA translation, since these proteins are involved in transporting mRNAs to specific cellular locations where they're supposed to be translated, either constitutively or in response to outside signal. For example, mRNAs that are stored in leading edge of epithelial cells, and only upon a signal, the proteins are actually translated in response to that signal, and the cells can migrate to where it's supposed to. There's also fascinating literature in the past year showing that many mRNAs are actually translated on the organelles where they're required to, for example, or on endosome or mitochondria, on lysosome, And then there's a whole machinery of protein that is responsible for transporting these mRNAs on microtubules in order to allow for this transport of the mRNA to the correct place. This is especially important in neurons, where many mRNAs are transported along neural projections, axon or dendrites, and they are stored at nerve endings, and only upon a signal these proteins are actually being made. In this area of mRNA translation regulation, there's a few potential ways to target translation, and there are companies that using target-based approaches where they try to directly target the RNA itself by looking for compounds that can directly bind to mRNA tertiary or secondary structure. Essentially, it adopts many different structural elements, much like proteins, and it is feasible that we will be able to identify compounds such as that. Another option is to target specifically a protein RNA interface, either by doing it computationally or doing in vitro-based assays and identify compounds like that. At Anima, we adopt I would say another modality where we use phenotypic analysis, where we can monitor the translation of target genes, and we do not a priori have a hypothesis on which one of these two potential elements is the most important for the disease situation. We have developed a platform technology which allows us to monitor translation in a high content manner using a microscopy. We have two collaboration with Eli Lilly and Takeda in the neuroscience field, and our technology is backed by 7 different patents. In the past, we had many scientific collaboration that showed that the technology indeed works, and we can monitor with it the translation of a specific mRNA of interest. Using our platform, we have developed a different or extensive pipeline targeting fibrosis, different targets in oncology, and different targets in neurosciences, and our most advanced project are close to IND enabling studies, both in fibrosis and oncology, and I will show you a few examples from this project right now. So this is the main theme behind our technology. We have developed a modality where we can label tRNAs without interfering with their function. We're labeling them as a thread donor and acceptor, and only when the two tRNAs are found on adjacent codons within the ribosome, a thread signal can occur. And essentially, this is what we measure. We transfect these tRNAs into cells, and we can monitor the thread signal coming from ribosomes in which these two tRNAs are sitting adjacent to each other. Because the human genome is known, we could essentially calculate all the codon pairs that are available in the human genome and calculate a signal-to-noise ratio for a specific codon combination, and our ability to specifically monitor the translation from an mRNA of interest. This is how the light looks like. This is coming from our fibrosis project, and what you can see here are thread signals coming from ribosomes, which are actively translating collagen 1A1 mRNA. We are always asked how selectivity can be achieved with this technology. So you need to remember that for our 20 amino acids in human, we have 46 codons, and there are more than 1,000 different combinations. And it turns out that many of our genes actually use codon combinations that are a unique signature for that gene. Calculating bioinformatically in silico all the potential combination, we find that 85% of human genes are actually containing a codon signature pair, which with a signal-to-noise ratio which is above 20, which is sufficient for us to monitor the translation of an mRNA of interest. This is essentially coming from our fibrosis project, just showing you the general idea on the collagen gene. So collagen has 300 different, 450 amino acids. 300 of these are proline glycine residues. In humans, we have 3 codons for proline, 3 codons for glycine, 9 different combinations. And it turns out that out of these 300 repeats of proline glycine in collagen 1, 120 of these use one of the 9 combinations. So even within the collagen family that is using proline glycine repeats within all of the 28 different genes, the collagen 1 preferentially use one of these codons. Other collagens either use one of the other 9 combinations or have no preferential at all. So overall, the signal we will monitor from collagen 1 using this pair will be highly specific to collagen 1A1. On top of that, it helps that not all 28 collagen are expressed in the same cell, only about 4 do. So basically the noise value is decreasing when we think of the mRNAs that are actually expressed in a cellular environment of interest. So these are the lights coming from quiescent fibroblasts that make steady state level of collagen. So we see very few spots of light coming out of the cells. But when we activate these cells into fully matrix deposition cells, collagen translation is enhanced by about 100-fold, and we see many spots of light coming from these cells, which is corresponding to a high protein accumulation 72 hours later. So this is 24 hours of induction. We can monitor this response very quickly. During a screen, we've developed a machine learning-based methodologies that essentially compare single cells in DMSO control wells with single cells in compound-treated wells, and essentially looking for anomalies between activated cells and compound-treated cells, and looking for these small differences between the cells. Because the analysis is unbiased, we're not looking for a specific change, let's say a decrease or increase in signal or number of spots, but just for any change, we are able in a single screen to identify compounds that reduce the signal or increase the signal or give any phenotype change to the signal we observe in DMSO control wells. Specifically in our collagen project, we were able to identify compounds that reduce collagen accumulation at the end of the day or vastly increase collagen production. To support this, the high content modality of our platform and enable a quick understanding of the mode of action and identifying the target for our project, we have also developed additional modalities to complement our screening assay, which is the translation light modality. And we have developed 2 modalities, which we acronym Pixel and Transcript light. Pixel is a collection of antibodies against key proteins that regulate transcription to translation. And this helps us to identify what the compounds are doing. For example, a compound that change splicing by changing the localization of splicing factors, a compound that affect ribosome localization, or compounds that change the localization of RNA binding proteins, which responds to specific stimuli. So using these type of assays with a library of known inhibitors, we were able to assign fingerprints to each one of these phenotypes. And when new compounds come in, we run in these high content assays and identify what the compound's mode of action is. In addition to that, we can follow the mRNA itself and essentially see what the mRNAs are localized in response to compound. So all of this helps us to identify the mode of action and the potential targets for the compound, RNA binding proteins, regulation of tRNA abundance, and any other of the potential targets that we may have. To support this, we have developed a cloud-based machine learning image analysis and data analysis crunching machine. And essentially the whole thing is automated. And at the end of a screen, we get score per compound and take it from there for a medchem campaign and compound optimization. I will show you an example from our collagen project. So going into the project, we use our bioinformatic analysis to generate a map of the gene of interest and identify the regulatory cis-regulatory element in the gene, which gives us an idea later on what the compounds are doing and what is a potential target. And we also in silico identify a list of proteins that may modulate and bind to these cis-regulatory elements. This is a general scheme of our campaign. We have a diverse compound library that we routinely use in our screen. As a primary assay, we use our pairs of tRNAs, and later on have additional secondary and tertiary assays that ensure that the compounds inhibit or regulate specifically the mRNA of interest and are not global mediators. This is how we validate that the pair of tRNAs are essentially showing us the translation of collagen 1. So this is the signal we're measuring with the pair, and with siRNA, as you can see here, almost 90% of the signal is reduced, indicating that most of the signal is coming from collagen 1 mRNA translation. In this project, we were able to identify 3 different chemotypes, and which were optimized to work in a nanomolar range, reducing collagen protein accumulation. And we show here those dependent images showing that the protein was reduced by each one of these series. Importantly, early on, we asked the question about tissue selectivity, and I took fibroblasts derived from lung, skin, or liver. These are all human cells. These are non-induced and induced cells. In green is the collagen induction, and we show that the compound from the 3 different series inhibited collagen production in lung, but not in skin or in liver. And this is just to show that everything is converted into data points and done in a dose-dependent manner, showing that the compounds indeed are very active in lung fibroblasts in orange, but inactive in liver and skin fibroblasts. So there's a very high tissue selectivity for regulation of collagen mRNA translation. In order to identify the mode of action of the target, the assay that starts the whole program is a Timecross analysis of the effect of compounds on the protein level, and at the same time looking at the mRNA levels using a collagen 1 specific probes. And what we seen using this modality is that a series 21 compound reduced the protein but showed no effect on mRNA, so we knew the compounds should be active at the level of translation. Cluster 7b, we saw that there's a small effect at the mRNA in the cytoplasm with no effect on a transcription site, so we suspected this is mRNA processing. In cluster 9, we saw destabilization of the collagen 1 mRNA in the cytoplasm, again, without affecting transcription, indicating that because what we knew from our bioinformatic analysis, that we are somehow disturbing the steady state level of the mRNA, and this is probably through poly-A site selection. Summarizing a lot of experiments we have done, we've identified the mode of action and target for each one of the series. Series 21, it's a protein that is involved in binding to the collagen 1 mRNA and keeping it in the cytoplasm and not being available to ribosomes. And when we use siRNA against this target, indeed we saw a reduction in collagen 1A1 protein translation. Series 9 is working by essentially inhibiting polyadenylation site 2 selection, which is something that happens after induction, generating only the use of polyadenylation site 1 and generating a protein with shorter half-life. Series 7b is interfering with co-translation insertion of the collagen nascent peptide through the ER, thereby inhibiting its production. We've also been able to translate the activity from our cell-based assays to animals using bleomycin-induced lung fibrosis. And as you can see here, we have successfully shown that the compound indeed completely inhibit collagen accumulation in this model, as indicated here. The lungs almost look normal like the naive animals, while nintedanib, which is used as a drug, an autotaxin inhibitor, which was in phase 3 studies, did not show such a robust effect on collagen deposition in the lung, and this is also shown here in the Ashcroft score. In our oncology project, we looked at the regulation of c-Myc mRNA translation, again, having a pretty good idea of which proteins are involved in this process by doing our expert analysis on the c-Myc pre-RNA. Using the same modality, we were able to identify a pair of tRNA and follow c-Myc mRNA translation and validate by SI that this is indeed what we are measuring. We also here identified 3 different compound series that in a dose-dependent manner completely inhibited c-Myc protein translation in cells without affecting global translation or transcription. Series 14, just as an example, reduced the protein levels and also reduced the amount of mRNA we saw in the cytoplasm without affecting transcription. And by using RT-PCR, we indeed showed that 2 active compounds from the series promoted decay of c-Myc mRNA as measured by RT-PCR, while an inactive compound from the series did not show this effect. Because we knew that this is mediated through the 3' UTR and we had a list of suspect RBPs, we used an siRNA library, and one of the RNA binding proteins, when we inhibited its expression, we also saw that c-Myc protein levels were reduced. Later on, we were able to show that this RNA binding protein is affected by the compound, and this RNA binding protein is binding to the c-Myc mRNA is enhanced, resulting in promotion of decay. Series 13 is working by transporting the c-Myc mRNA into RNA granules, which are protected from ribosomes. And series 4 is working via polyadenylation site selection, and again, generating a shorter half-life c-Myc mRNA. Here too, we were able to take series 14 forward and show that it was able to inhibit tumor growth in A549 xenograft models. Almost complete inhibition was seen, and also protection in mice survival. In our neurobiology project, we look at tau protein, and using exactly the same modality, monitoring tau translation, we were able to identify 2 different series that inhibited very efficiently in a dose-dependent manner the tau protein in the initial primary assay neuroblastoma cells that we've used. And also recently, we've shown that using primary human-derived iPS neurons, the compounds were able to very efficiently inhibit 300 nanomolar, this is an optimized hit, tau accumulation without affecting neuronal health, which is measured by MAP staining and DAPI staining. The mode of action of the compound we've started to look at, and it turns out that series 418 results in, again, promoting the decay of tau mRNA in the cytoplasm and retaining the mRNA in the nucleus, while series 12065 is not affecting the mRNA at all and probably works at the level of recruitment of ribosomes. I want to thank you for the time, and I'm open for any questions you